test that we do in IMR. Then we will discuss on the future and emerging technologies. So a bit on the history. In 1994, Winthrop is a famous hematologist at that time. He stated that no cause for leukemia was known at that time, and instead he described diseases and infection were found at the leukemia at diagnosis. So in 1953, then the structure of DNA was understood by Watson and Crick. And in 1960 is the big discovery for hematology in the world, where Philadelphia chromosome was first found. It is a chromosomal abnormality in CML, which is the landmark as the first consistent abnormality found in any cancer. And in 1993, Kerry Mullis received a Nobel Prize in chemistry for his invention of the polymerase chain reaction. So then the molecular work starts. So these are the evolution of molecular diagnostics in hematology. We start with in 1900, we just look at the morphology through microscopy. Then in 1960s, the karyotyping, followed by immunohistochemistry. This is from the bone marrow aspirate and trypan biopsy. We do immunohistochemistry. After that, the PCR and FISH. In 1990s, the Sanger sequencing was found, and then followed by microarrays, and then in 2010, the next generation sequencing. So before we go further, I will go generally on the hematological disorders. Actually, there are a wide spectrum of hematological disorders. So we just stop on the hemato-oncology, mainly the cancer, but we also have some benign type of cancer. And then we have red cell and membrane disorders such as thalassemia, the membrane disorder, or the enzyme deficiency such as G6PD. And we also have a group of like bleeding disorder and thrombosis such as hemophilia A and B, the von Willebrand disease, and also thrombosis such as protein C and protein S deficiency. So I'll give example on the workflow for lab diagnosis in acute leukemia, the real life what happened in the ward and the hospitals. So usually when the patient come to the hospital, leukemia patient, they usually come with, they are usually very ill, they are anemic, they are pale, and some will come with bleeding and infection because the white cell is very high, and the other parameters, the hemoglobin and platelet, is low at that time. So what usually happen in the ward, the doctor will do full blood count. So what we will find usually, I think you all know that they will find the high white cell count. So then usually we will do full blood picture. So usually this is what we can see in the full blood picture. We can see these are the leukemic cells, or we call it blast cells. So after that, we need to do bone marrow aspirate and trypan biopsy. So these are some of the findings of the bone marrow and trypan biopsy. I think you all already learned on this. These are the example of the spatial stain need to be done, the immunohistochemistry on the BM80. So this usually happen in hospital, and I believe all of the hospital we are able to do this step. After that, when they do bone marrow aspirate and trypan biopsy, we will take extra samples that we will send for immunophenotyping, cytogenetic, and also molecular. And these tests are only available at the referral center. In the KKM hospital, mainly it is in Hospital Tunku Azizah, the Women and Children Hospital, and for immunophenotyping and cytogenetic, and molecular, usually they will send to IMR or Ampang Hospital. So I will touch a bit on the immunophenotyping or the flow cytometry, because for bone marrow aspirate and refined biopsy, I think it's under the basic test. So what is flow cytometry? It is a laser-based technology which is able to identify and quantify cell populations. These are the FACS Canto, the machine that we use, and usually in normal cell, it has a characteristic profile, but malignant cells, they usually express an aberrant phenotype, the abnormal phenotype that can be useful for their detection. So what we do, usually we will lyse the cell and stain with antibody label with different fluorochromes, and this is to recognize the pattern and intensity of expression of different antigens on the surface of normal and leukemic cells. And then this cell suspension manipulated into the stream of fluid. So where the single cells are interrogated by electronic detection system. So we will identify the cells, and these are the real life, what happened in the machine. So these are what we get. This is the analysis of acute lymphoblastic leukemia T lineage. From using flow cytometry, actually we already can diagnose the acute leukemias. Yeah. So these are the forward and side scatter. So this population is actually the blood cells. So what we can see here that these cells are positive for cyto-CD3 and NTDT, which is the immature marker and a marker for T cells. So here it is also positive for CD2 and 34, also the marker for immaturity. And it is positive for CD7 and also CD3. So this actually, with this knowledge, we can diagnose that this patient has T acute lymphoblastic leukemia. So what's next? Now we move to the advanced test, the cytogenetics. This test is actually not so much of the diagnosis. It is more on the prognostication. So actually the diagnosis can already be made by the bone marrow aspirate and refined biopsy and also flow cytometry findings. So, but some cytogenetic findings can help for diagnosis. For example, this Philadelphia chromosome. So what is cytogenetic? It is a classical karyotyping of chromosome with the resolution of more than 3 to 5 MB. It remains the most comprehensive method for assessing the chromosomal abnormalities, especially the numeric and structural chromosome aberrations. So it is essential for disease classification and prognostic assessment and also treatment decision. And here is actually the Philadelphia chromosome. This is the famous translocation 922. The first chromosomal abnormality found in cancer. So these are the cytogenetic techniques. Just give the overview. What we do in the lab usually, we will first do the bone marrow culture overnight, and then we will harvest the cell, and then we will do slide preparation and staining, followed by microscopy and karyotyping, image capturing, and also reporting. This is the conventional method. Now it can be replaced by the Meromax or the this is the commercial. This is the commercial kit, which can reduce the time for the activity in the lab. So what we can see here is the metaphases that we can see under the inverted microscope. You can see here the chromosome. And then this is the analysis of metaphase spread, what it looks like under the chromosome and under the microscope. And after we capture the the image, then we will have to use the software to arrange this, do the karyotyping, and we need to arrange accordingly. And this require a very skillful person to do this. Okay, this what happened in the lab. We received the cytogenetic request from the hospital. These are the samples, how they process in the lab, processing of the bone marrow sample, do the chromosome spreading, staining, and then the slides are ready for analysis. They will load into the scanner, and here are the staff doing the analysis and karyotyping. So this is the automatic cytogenetics platform cytovision scanner for image capturing, and it can also be used for fish scoring. So what are the advantages of cytogenetic? It is able to detect additional cytogenetic abnormalities which cannot be detected by local specific techniques. For example, the FISH or PCR, which I will explain later. Disadvantages, it cannot detect small deletion of UKB pairs length and cryptic translocation. It is laborious, and it also have some technical issues which resulted in the increased use of other techniques such as FISH. However, this cytogenetics still remains the gold standard for most numerical and large structural chromosomal abnormalities. So now we move to other technique, which I categorize under diagnostic molecular techniques, the FISH, array CGH, PCR, and also DNA sequencing. So what is FISH? It is a fluorescent in situ hybridization. It is a molecular cytogenetic technique which identifies chromosomal abnormalities using molecular technology. It uses fluorescent probe which hybridize to the complementary sequence and results in the signals, and then we need to examine under a fluorescent microscope. So this is the principle. So the probe DNA with a specific sequence is labeled with fluorophores, and the probe and target are denatured, usually using heat, and then the temperature is lowered to allow the probe to reanneal or hybridize. And then after washing away non-specific or excess probe cells are counterstained to show the chromatin location, and observe on a fluorescent microscope to determine the location of the probe signals. So we have few types of probe, the dual color dual fusion probes, break apart probes, the deletion duplication probes, and centromeric probes. So here how it looks like, the FISH interface. You can see here is actually the BCR-ABL1 translocation detected using the BCR-ABL dual fusion probe. You can see here the two red-green fused signals of the ABL and BCR genes, and these are the two signals for each color in normal cells. You can see here, you can see the fusion of ABL and BCR genes. Now we move to the array comparative genome hybridization, or array CGH. This is a technique that was developed for high resolution genome-wide screening of segmental genomic copy number variations. It has high resolution of 30 kb. The clinical utility is for detection of genetic abnormalities in leukemia and lymphoma. However, it is usually used as research, and it is not used in diagnostic yet in our country. And this technique is actually widely used in the diagnosis of genetic abnormalities in children with developmental delay or intellectual disability or autism spectrum disorder. What happened? In this test, the equal amounts of labeled genomic DNA from a test and a reference sample will be co-hybridized to an array containing the DNA targets. However, it is compared to cytogenetic, this test, even though it can give high resolution, it is unable to detect balanced translocations. So here we can see the process. The patient and control DNA are labeled with fluorescent dyes and applied to the microarray. You can see here. And patient and control DNA compete to attach or hybridize to the microarray. And the microarray scanner measure the fluorescent signals, and then the computer software analyzes the data and generates plots. Here is actually what it looks like. Here is the scanner. The actual array is like this, where we can detect duplication and deletion, and these are the array profile. So these are the examples of genomic imbalances detected by array. You can see we can detect very small gain or loss in the chromosome. So if we compare for karyotyping, the genomic resolution is 4 to 5 MB. For fish is 40 to 250 KB whole genome, and chromosomal microarray is 30 to 30 KB. But for fish, it's actually because it is you need to know the exact location and use specific probe. And then we move to PCR. PCR is a molecular technique used to amplify a specific region of DNA sequences of interest, and it is the most commonly used molecular assay in the management of hematological malignancies. And the main component in PCR is the DNA template, the primers, forward and reverse primer, tag polymerase enzyme, the nucleotides, and also magnesium. So PCR procedures involve 20 to 40 thermocycles, which is comprised of denaturation, annealing, and elongation in each cycle. So after we do that, we need to do gel electrophoresis. This is actually the post-PCR analysis step. Here, the PCR fragments can be identified through size separation of the PCR products on agarose gel electrophoresis. So now we move to RT-PCR, the reverse transcriptase PCR. It is the commonest type of PCR used in practice today, especially in hemato-oncology. It uses the enzyme reverse transcriptase to synthesize what is called as cDNA or complementary DNA from mRNA, the messenger RNA. So the process is also similar from what I've shown earlier, but we just use this enzyme reverse transcriptase. So the final amplified product is assessed by agarose gel also, and here we can study the expression of genes in a qualitative manner. The other type is real-time PCR. So in real-time PCR, the amplified DNA is tagged with non-specific fluorescent dye that intercalate with any double-stranded DNA. So then the thermocycler, the machine, which contains sensor for measuring of fluorescence emitted from the fluorophore after it has been excited at certain length. And generation rate of fluorescence is measured, and real-time values of DNA amplification are provided. Here that we can see the real-time amplification of the DNA. So this technique can quantify copy numbers of a given DNA sequence in a sample. So these are the machine that is used, the real-time PCR machine. And this technique is commonly used for specific gene detection. For example, the minimal residual disease monitoring in PCR APL1 and PML-RARA. This is the important gene fusion that need to be monitored. Now we move to Sanger sequencing. This technique is first introduced by two-time Nobel laureate Frederick Sanger and his colleague in 1977. Here, the chain termination method by fluorescent label ddNTPs by DNA polymerase during in vitro DNA replication is used. It is a post-PCR analysis and a confirmation method for mutations. So when you want to do Sanger sequencing, we still need to do PCR first. Then we will do Sanger sequencing. So what is happening during the cycle sequencing? First, denaturation to separate DNA into two strands, then annealing, the primer binding, elongation, addition of the NTPs to form new DNA strand, and then chain termination, where ddNTP is incorporated into the growing DNA strand and thus stop the process. So how it looks like? I think that that I have explained earlier. Okay. So the of target gene product. The second step is that we do cycle sequence PCR with a chain terminator fluorescent dNTPs. And then after that, we can detect the products with capillary electrophoresis. This is the electropherogram generated. So the sequencing analysis then is done by software. It provides analyzed sequence. This is what we call the electropherogram, and peak patterns reflect the mutations or sequence changes. This is the heterozygotes. Okay, after describing all these tests, this is comparison on the sensitivity of the test. So by morphology, which is by when we view the blood cells under microscope, this is the sensitivity. It's 1 cell in 10. So you can compare that with cytogenetics, it increases, and PCR is give the highest sensitivity. So now we move to the clinical applications of molecular techniques and genetic markers in hemato-oncology. So it helps in the initial diagnosis, for example, the detection of fusion genes and also detection of mutations. It can also help the clinician to establish treatment protocol by giving the information for risk stratification and also prognostication of the disease. Other than that, it also helps in monitoring the response to therapy, what we call minimal residual disease. And for some leukemia patient, they usually need to go for transplant. So we can use this technique to monitor post-transplant chimerism analysis, which I will show later, and also to determine the lymphoid clonality for some cases of lymphoma or acute lymphoblastic leukemia. So this is just to share with you for the doctor in the hospital and also for the pathologist, when we do diagnosis, we base on this WHO classification. So actually recently, I think it's in 2016, they have moved to integrate or give a closer integration of morphology and genetics. Previously, we focused more on morphology of the cells. Now we already include the genetics for the classification. So by these updates, they generate new diagnostic approach and improve prognostic and predictive models and also therapeutics, because with current finding of genetic abnormalities in the disease, we can go for targeted therapy. So this is called the precision medicine, why they move from just morphology into molecular and genetics. This is just to show you that different level of integration of genetic data into clinical pathological classification of hematological malignancies. For example, the diagnostic criteria of, we use JAK2 mutation in myeloproliferative neoplasm. For TP53, if it present in a patient with AML, it give bad prognostic factor, and we can also look at the predictive factor of BCR-ABL1 in ALL, and some disease-defining lesions such as 5q deletion in myelodysplastic syndrome. This is just to share how they incorporate this AML. This is actually, I just focus on acute myeloid leukemia. They now come up with new category, the acute, the AML and related precursor neoplasms, where they incorporate one group called AML with recurrent genetic abnormalities. This is just to show that how the diagnosis evolve. I don't think you need to remember all this, just to show you the different type of AML. This is also how the molecular classes of AML and concurrent gene mutations in AML occur. And here is actually the risk stratification by genetics by European Leukemia Net 2017. This is actually important because the clinician would like to know the genetic abnormality, because then they will know whether this patient has favorable or intermediate or adverse prognosis. So from there they can have an idea to plan on what type of chemo or what type of therapy to give to the patient, whether to escalate the dose or decrease the dose based on their prognostic factor. So now we move to the molecular hemato-oncology test offered in our lab. Yeah. So in our lab, we have leukemia translocation study, the AML and CML mutation study, and also chimerism analysis. For non-hemato-onco, we also have thalassemia and hemoglobinopathies. So I will also touch briefly after this. So this is actually our request form if you want to send sample to our lab, or maybe one of us will join us later after you graduated. So this is actually algorithm in our lab. When we receive specimen for hemato-oncology, we will first do extraction. First, we will do RNA extraction. Then we will do the translocation study. And if the patient has acute myeloid leukemia, we will extract their DNA and then proceed with this AML mutation study. We follow this algorithm to determine what is the next step to be done. So for our leukemia translocation study, we use commercial kit which can identify 30 chromosomal translocations, and we use the real-time qPCR techniques that I've shown you earlier. And we started this test in 2008, and we offer for the whole country in 2012 until now. So these are just the list of the 30 leukemia translocations that we do in our lab. And this is our old data, about 10 years data, which we have about 4,400 patients. So you can see here that we found about 45% patient with BCR-ABL, the 1221, 1517, and 821. So now we move to the acute myeloid leukemia mutation studies. So for acute myeloid leukemia, the management of patient really relies on the genetic test, because it establishes the diagnosis, prognosis, or prediction of response to therapy. So molecular mutations in AML have been analyzed to subdivide AML into prognostic subsets, which I shown earlier, and we offer this test since 2013. So in our lab, we offer 4 tests: the FLT3, NPM1, c-KIT, and CEBPA mutation, using various techniques. The FLT3 we detect using PCR and restriction enzyme digestion. For NPM1 and c-KIT, we use real-time PCR, and for CEBPA, we do PCR and sequencing, and we offer this since 2013. Next is chronic myeloid leukemia mutation study. This I just show how a patient with chronic myeloid leukemia, they usually have dysplenomegaly. Characteristically, they have a very big spleen. So with the discovery, and usually the CML patient, they have this 922 translocation that I've shown you a few times before. And discovery of this tyrosine kinase inhibitor called imatinib mesylate in 2001 has resulted in its use as a frontline therapy in CML. However, the long-term use of this medication results in emergence of therapy resistance. So usually patient, when they take this medication, they will become better, almost normal, and their big spleen will shrunk. So it gives really tremendous life-changing event in their life. So however, with prolonged use, they may develop resistance. So we need to identify the type of what happened, because when they develop resistance, this medication cannot function normally. It cannot bind to the specific BCR able target. So we need to do this test because usually 30 to 50% will have one or more BCR able kinase domain mutation. So we will do BCR able 1 kinase domain mutation using PCR and Sanger sequencing. Okay, this test actually offered since 2014. Previously, we just detect one type of mutation, the T315I mutation, the most severe type. But now we able to look at the whole spectrum of the BCR-ABL1 kinase domain, which is actually the gold standard for this test. So this is actually just to show you the BCR-ABL kinase domain. So actually, according to literature, we identify actually more than 100 type of mutation. So using this technique, we are able to determine, we are able to identify this mutation if it occur there. And usually, this information is very important for the clinician, because if they know what type of mutation, so they will change the medication from, because imatinib is the first line, they might change to second or third line, such as nilotinib, dasatinib. They have lots of medication. So next, in our lab, we also do chimerism study. So what is chimerism study? Chimerism study is actually done for patient who undergo transplant. So it is a measure of the number of donor and recipient cells in the host following hematopoietic stem cell transplantation. You can see here, what if you familiar with the word chimera. Chimera is actually the single organism. It's a Greek word. It is a single organism that's made up of cells from two or more individuals. So for patient who undergo this post transplantation, so they actually have other people's DNA, because their body is actually, when they have leukemia, their body is actually full of leukemic cells. So when they give chemo, we don't want their, if we aim for complete cure, we don't want their bone marrow anymore. So we will give chemotherapy and radiotherapy. So we can imagine that their marrow is empty, and then we will give other people's or the donor bone marrow, the stem cells, and then there will be like a new people, that's why they will have a different DNA in their body. So if we test their the buccal swab is different from their bone marrow DNA. So this test can be used for prediction of disease relapse, graft rejection, and graft versus host disease. So in IMR, we use commercial kit using real-time qPCR technique. So we offer to HKL since in 2010. Then we offer to other transplant centers. There are not many transplant centers in the country, so these are among the transplant centers. So other than hemato-oncology, we have other diseases as well. So in IMR, we do molecular diagnosis in thalassemia and hemoglobinopathies too. The main techniques is mainly PCR. Here we use multiplex gap and arms PCR. It's slightly different in terms of the design of the probe, because for multiplex gap, the aim is to detect gene deletion, but arms is for detection of point mutation. For complicated case, we do MLPA, and for an unresolved case or for rare mutation, we will perform Sanger sequencing. So just a brief overview for alpha. We have, as we know, we have alpha and beta thalassemia, and I believe our students here maybe during their Form 4, they already have their blood taken for thalassemia screening. So for those with abnormality or alpha thalassemia, we do the PCR for do genotyping for them. So for alpha thalassemia, the most common abnormality is deletion. An individual with thalassemia may have will have one alpha gene deletion, we call alpha plus, or two gene deletion, we call alpha 0. So these are the common abnormality found in our population. So we can also have non-deletional forms, or we call it mutations. Oh, this typo here. And in our population, actually, we have more than 30 mutations, but the common one in Malays are the HB constant spring, and this is detected by this PCR technique. So this is how the multiplex gap PCR for alpha thalassemia looks like. These are all the patients. Next, we move to the beta thalassemia. It is mainly caused by point mutations. For alpha, if you can remember, it's deletion, but for beta, it's mainly point mutations. So the detection is using ARMS PCR. But if we cannot find using this panel, we will continue with the DNA sequencing. So these are the ARMS PCR for our beta thalassemia patient. These are among the commonest abnormality in our population, the IVS G2T and 28. So these are just to show you, we have lots of panel for our alpha thalassemia, our alpha gap arms. We can detect different type of deletion, mutation. This is for MLPA to detect deletion of unknown region, and then we also do sequencing for our complicated cases that we cannot find using this technique. We also do for alpha triplication, and we have special ARMS technique for this mutation. This is for beta globin gene genotyping. Mainly, we use ARMS, multiplex ARMS PCR to detect this common mutation in our population. If we cannot find, then we will do beta sequencing. And if you know that apart from thalassemia, we also have hemoglobinopathy, such as hemoglobin E, hemoglobin C, hemoglobin S, or sickle cell disease. So for your information, in our population, we have a lot of patients with HBE. So we design a special ARMS PCR for this, the HBE, HBC, SD. And gap is also for deletion, even though it is rare for beta thalassemia. We also have some rare deletion, so we also have the panel. This is also the hemoglobinopathy, the HB Lepore, and also sequencing. Okay, now we move to the future technique. The next generation sequencing, it is the application of high throughput massively parallel sequencing technologies. We have many types of NGS. The first one is whole genome sequencing. So for whole genome sequencing, it provides a comprehensive overview of the entire 3 gigabases human genome at single nucleotide level resolution, and it covers both coding and non-coding regions. For whole exome sequencing, it analyzes the protein coding sequences of the genome, and the sequencing is restricted to exonic regions of the genome where the pathogenic mutations are more common. And for targeted sequencing, it allows for the selective arrangement of particular genes of regions of interest. And this is actually usually more cost effective, and it is ideally used in the diagnostic lab for the evaluation of malignancies where the spectrum of mutations is well established and the discovery work is not required. For this whole genome and whole exome is usually more suitable for research, and this for diagnostic targeted sequencing is more suitable. So this is just a figure to show the whole exome sequencing, the size and whole genome. So I just show for the targeted sequencing, it allows for the selective arrangement of particular genes of interest and ideally used for diagnostic lab. So we start with the sample preparation, the genomic DNA extraction, and then the amplicon library constructions. Then we use targeted NGS panel. For example, we have many types of commercially available panel with multiple numbers of genes, and then we need to put into the sequencer, and this is how the data, and then we do variant calling and analyze the data. And for NGS, we need bioinformatic analysis for it is not easy and not straight to the point to analyze, where we need the bioinformatic knowledge to analyze this data. So the next is imaging technologies. This is actually the last part. I just want you to know about this. So what is liquid biopsy? This is actually increasingly being studied because it is a potential tool for refining diagnosis and disease monitoring. It is called term liquid biopsy, which means we can assess tumor DNA through just simple blood sampling without the need of an invasive tissue biopsy. Because, for example, patient with lymphoma, they have big lymph node, so we need to poke them to get the samples. But this liquid biopsy, we can detect disease just through simple blood taking. So for example, in cancer, in hematological malignancy, we can detect this through circulating tumor DNA, because in our body, if the cells within the body die, they are broken down. So components of the cells, which is very small DNA fragment, we call it cell-free DNA, are released into the bloodstream. This is the concept. So we can isolate this cell-free DNA by extracting it from the plasma. And if the patient has cancer, we can detect a subset of this cell-free DNA, which is called the circulating tumor DNA. So what we can do with this clinical application is that it can establish the molecular fingerprint of the tumor at diagnosis, or if when we do lymph node biopsy, the tissue is insufficient or cannot be sampled, or kalau patient is too ill for biopsy, or at the time of progression of the disease, where we can find some resistant mutation. And we can also use for confirmation of targeted therapy and mechanism of drug resistance. We can use for monitoring of disease, early detection of metastasis, and it can also provide prognostic information, and it can complement tumor molecular profiling before, at the same time, or after the tumor biopsy. I think we come to the end. So as a conclusion, molecular technologies and understanding of genetics have introduced new insight to hematological diseases. And advances in molecular genetics will generate new diagnostic approaches, improve prognostic or predictive models, and enable targeted therapy for precision medicine. I think with that, I thank you.
Right. So thank you, Dr. Srina, for the very comprehensive and excellent presentation. So I think all of us now know all of us know how to sorry, there's a problem here. Okay. So I think all of us already know the current or the advanced techniques available out there for the detection of or for the diagnosis of hematology diseases. Okay, so as you can learn from the presentation just now, there are a lot of testing, okay, for the hematology diseases, but most of it is molecular techniques, which includes the PCR and also NGS, which is the new technology. So in the era of precision medicine, as mentioned by Dr. Srina, I think we need to take advantage of the development of technology with the introduction of many molecular testing so that more definitive diagnosis can be made. Okay. All right. So before I open for Q&A session to participants, I would like to ask one question to Dr. Yusrina. Okay, Dr. Yusrina, I was wondering, so there are a lot of specialized testing available. So how does the lab decide which test to be performed for specific diseases? For example, AML. So how is it based on the request from the clinician or is it decided by the lab itself? Because I, from my conversation with my colleague in hospital, sometimes they are not aware with the available tests, right? So does IMR also provide some recommended testing for them?
Okay, for the test, usually we will engage with the clinician, and since this test is usually very high-end test, and this patient, usually with the hemato-oncology patient, they will be sent to specific test with the specific hospital with hematological services. So I think it's may not be known to the basic the primary hospitals, because usually the test usually ordered by the clinical hematologist, which is they are located in the center. So maybe they some of them do not aware of the test. But usually when we want to offer the test, we will communicate with the clinical hematology service.
All right. So does the clinician use that kind of information for the testing?
Yes, because for the leukemia translocation study, for example, they really use it for prognostication. And for example, there are among all of that, most of the leukemia translocation study is for prognostication, but two type of translocation which they usually want to know as soon as possible. Usually we will need to give it within 3 days, the BCR able one and also the PML RARA, because this type of leukemia for PML RARA is a very life threatening, because patient can die anytime. So they need to give one type of medication. If this patient, like they have 15, 17, they will need to give specific medication called ATRA, the all transretinol acid, so that usually if patient given it this medication early, they will survive. And for BCR able one, it is actually associated with poor prognosis. And when they identify this, they will give the tyrosine kinase inhibitor, the which I explained earlier, the imatinib, in the management of the patient apart from the chemotherapy.
All right. All right. Thank you, Dr. Yus. So I would like to open the Q&A session to all the participants. So is there anyone want to ask question to Dr. Yusrina? So I hope students take advantage of this opportunity because it's difficult to get Dr. Suna because I know she was very busy. Okay. Although now with PKP, but I think the frontliners such as
Yeah, I'm in the office.
yeah, stay in the office. Can see the office there. Any question from the audience or participants?
It may be quite high-fi for the student, but I think it's a good knowledge. And when you join the service later, you'll be, hands-on.
Assalamualaikum, Doktor.
Salam.
Hello.
I want to ask about um, who are usually in charge for the a chromosome arrangement or the karyotyping? Is it the MLT or there is specific person to do it?
The test will be performed by the MLT under supervision of our scientific officer or research officer in the lab. But the trained technician can do the karyotyping. They can arrange the chromosome, but usually it is also done by our medical doctors, our scientific officers, and also the pathologists. And then but the final report will have to come from the scientific officer and also the pathologist.
Okay, thank you, Doktor.
Any more question? You may also ask in Malay if you're comfortable with Malay.
Ya, boleh je.
Yeah.
Doctor, I have one question. Between liquid biopsy or tissue biopsy, which are most preferred?
Which are most
Preferred.
preferred? Actually, tissue biopsy is still gold standard because we get the sample directly from the tumor itself. But as I said earlier, liquid biopsy is actually imaging technique. Many literatures and many researchers are looking into this. They try to compare liquid biopsy and also tissue biopsy to see whether it can give similar results. And many of the literature said that it can give similar results. So they hope that this liquid biopsy can actually we are at this stage we cannot say to replace lymph node biopsy, but may complement lymph node biopsy. But so far many studies shows that it can give similar result actually.
Okay, thank you, Doctor.
Right, so I think you may also ask, is there any potential opportunity to work in IMA later? So Doctor, so this may out of topic a bit. So the student here are they're already in year 2, right? Year 2? Semester 4, right? So they still have 2 more years to go. So what are the prospects for them to work in IMR? What position available for them?
I think for the students, they are degree in medical lab technology, right?
Yeah.
So I think they have the chance to go for scientific officer post or research officer post. So I think after you finish your study, you may apply through JPA for this post. And insyaAllah kalau ada rezeki boleh join IMR.
Lagi soalan?
Doktor, saya nak tanya.
Ya.
Kalau dalam lab kita untuk buat test kita ada algorithm kan? kita buat test tu based on severity patient ke, ataupun memang kalau kita receive sample kita follow algorithm test tu?
Oh, usually kan when you receive sample, kalau hemato-oncology, the pathologist or the doctor will screen the request form. So we will order what to proceed. And then the officer will look again, and also they will perform the test, and what happened in the lab bila kita dah buat results, all the technician do the results. So the officer, macam you all akan jadi the officers lah nanti kan. So the officers akan validate the results, prepare the report, and then bagi dekat pathologist will check again whether okay ke tak, teknik tu betul ke tak, and then sesuai tak correlate with the disease, and then kita sign off lah, baru kita dispatch the result. Macam tu dia punya flow dia. So macam you all, kalau you all join us as scientific officer, that's ni lah, you all have to oversee the test and do validation of the test.
Terima kasih.
Okay, ada soalan lagi? Mungkin 2 atau 3 soalan lagi. So how about Faiz, the class rep? Faiz, where are you Faiz? Faiz ada tak?
Hello, Assalamualaikum.
Salam.
Betul, saya nak tanya berkaitan dengan lymphoma. Saya nak tahu advanced lab test yang guna sekarang. Sebab saya baca kat Google macam confused lah juga. So banyak.
Oh, lymphoma dia memang complicated sikit. Dari segi lab test tu, dia buat masa ni dia banyak through yang immunophenotyping yang macam I tunjuk awal tu, flow cytometry tu kan. So yang tu kita, daripada situ kita boleh, sebab lymphoma ni dia satu disease yang sangat besar, so nak diagnose dia memang susah sikit compared to macam leukemia. So menggunakan flow cytometry tu kita boleh tahu dia punya surface marker, jadi kita boleh kategorikan dia ni jenis apa-apa, sebab dia ada lymphoma yang tak bahaya sangat, maksudnya yang benign, dan ada juga yang malignant. Tapi satu lagi, lepas kita, dia lymphoma ni kan dia banyak berlaku dekat lymph nodes tau, so dia boleh ada lymph nodes kecil atau lymph node dekat dalam badan yang kita tak nampak, so that's why dia kena ambil sampel. Tapi macam lymphoma, kalau dia dah banyak sangat dekat lymph node tu, dia boleh pergi masuk dalam the whole body, dia boleh pergi dekat bone marrow. So bila patient lymphoma, dia memang wajib kita buat staging bone marrow. So maksudnya lepas dia dah ambil biopsi semua tu, dia akan buat pula staging, dia akan buat juga bone marrow. Dan beza dia daripada bone marrow orang acute leukemia ni, dia kena buat dua belah tau, dia kena cucuk belah kanan dengan belah kiri dia punya tulang pinggul, sebab nak tengok, sebab sekarang kita nak buat staging, kita nak tengok dia dah pergi tak dekat bone marrow. Sebab bila dia dah pergi kat bone marrow, dia akan staging dia jadi tinggi, dia akan jadi stage 4 macam tu. So itu, dan bila kita buat bone marrow tu, kita akan guna pakai yang saya kata tadilah, bone marrow tu kita akan guna immunohistochemistry tu, yang staining tu kita akan tengok untuk confirm the diagnosis. Macam tu. Tapi sebenarnya diagnosis untuk macam bone lymphoma ni, dia lebih kepada tisu biopsi tu lah. Tisu biopsi tu sama jugalah kan, kita akan letak dia paraffin embedded, kita buat slide yang macam histo slides tu, tengok bawah mikroskop, and then kita boleh stain dia juga. Oh, jelas tak? Dia memang complicated sikit lymphoma ni.
Jelas. Thank you.
Terus apa lagi? So, boleh tanya lah macam Haikal, saya tanya regarding assignment kan. So you punya assignment
lymphoma
So
dia memang susah sikit lah, mencabar.
Assignment memang title pasal specialized test ni. So advantage lah, cuba tanya Dr. Yus.
Dia buat on apa? Tajuk dia?
buat tentang lymphoma lah. Macam
That means
general. Saya buat pasal advanced lab test. Jadi saya baca-baca confused.
Dia memang complicated sikit lah. Tapi apa-apa pun dia akan start daripada tissue biopsi. Tissue biopsi, lepas tu kita akan buat slide, boleh tengok dekat slide, and at the same time kita akan buat bone marrow, buat staging macam saya cakap tadi. Bila buat bone marrow pun samalah, kita akan tengok smear, lepas tu kita akan kita akan letak immunohistochemistry stain, and sebahagian daripada bone marrow sample tu kita hantar juga flow cytometry. Macam saya kata dia masuk kat mesin tu kan, dia boleh tengok yang gambar sel tu adalah sel kategori apa. Macam itulah dia punya diagnosis. Dan dia ada jugalah panel-panel yang kita buat juga cytogenetic, kita fish panel, semua tu boleh bantu. Tapi untuk lymphoma panel, kalau molekular ni dia tak ada banyak lah. So far kalau kat Malaysia kita tak offer, tapi kat overseas dia ada lymphoma panel yang guna PCR semua tu. Tapi kat Malaysia so far kita tak buat. Dia belum sampai that level.
Doktor, saya nak tanya, sekarang ni out of topic sikit, tapi sekarang kan tengah pandemik, so dia delay ke dalam proses all this, macam siapa yang hadap cancer, macam maybe nak ambil biopsi semua, dia jadi delay ke dia punya treatment, testing punya
Dia
proses?
in general sebenarnya semua akan affected lah, tapi macam kanser ni memang tak akan didelaykan lah. And then macam kanser bila masuk ward tu dia macam benda yang tak boleh delay kan. Tapi untuk testing pun so far kalau kat hospital, kalau yang benda-benda kanser macam leukemia semua tu memang tak affected lah. By right memang kita orang akan buat terus. Tapi benda-benda yang benign macam saya kata tadi, benda yang macam thalassemia kan, penyakit genetik, yang tu boleh macam melambatkan sikit. Tapi kalau benda-benda kanser memang tak boleh tunggu. That's why macam ni walaupun saya tak adalah kerja directly dengan COVID, tapi we all still kena datang kerja macam biasa sebab kes-kes leukemia memang kita tak delay.
Okay, thank you doktor.
Okay.
Ada lagi soalan? Mungkin last question, atau mungkin nak tanya pasal penyakit ke, family dapat penyakit kanser ke, mungkin boleh ambil kesempatan ni nak tanya Dr. Yus. So Dr. Yus, so seperti kita tahu kanser ni terutama sekali kanser leukemia, lymphoma kan yang berkaitan darah ni. Kita tahu dia treatment dia chemo. Kan, standard treatment dia chemo. Tapi majority patient akan relapse. Kan? Apakah bermaksud standard treatment kita untuk leukemia ni sebenarnya tak bagus?
Itulah saya rasa sebenarnya dia masih ada ruang untuk penambahbaikan. Macam saya rasa Dr. Fadli pun lagi arif kot pasal benda ni sebab macam bila seseorang patient tu dia ada dia dapat kanser, biasanya dia ada mutation, dia ada translocation, lepas tu dia ada driver mutation, cooperating mutation, benda-benda tu lah. And bila dia dah diberi chemo, kemungkinan besar dia akan develop sebenarnya mutation lain pula. So that's why itu yang mungkin akan contribute dia untuk relapse. Dan mungkin juga treatment tu tak betul-betul macam bunuh ataupun semua leukemic cells tu, dia ada residual lagi.
Ya, ada lagi soalan terakhir lah, soalan terakhir pada partisipan sebelum kita tutup. Ada soalan daripada lab staff tak?
Lab staff ada juga
Lab staff ada juga. Syaraf ada juga. lah. Saya rasa tak ada doktor semua dah faham. Okay, baik. So, terima kasih Dr. Yusuna. So, bagi pihak Pusat Pengajian MLT UiTM, saya mewakili semua pensyarah dan pelajar serta staf Makmal mengucapkan terima kasih di atas perkongsian Dr. Yus. So, saya rasa semua dah sedar yang terdapat macam bagi pelajar terdapat banyak lagi teknik yang ada kat luar sana. So tujuan webinar ni adalah untuk mendedahkan mereka untuk dengan teknik lain apa yang diorang belajar dekat UiTM ni. So saya rasa objektif webinar ni tercapai dan saya rasa itu saja daripada itu saja untuk webinar kali ini. So, semoga jika ada peluang di masa depan kita dapat berkolaborasi lagi sama ada dengan next webinar ataupun mungkin latihan dan sebagainya.
Okay.
Terima kasih Dr. Yus.
Okay, sama.
Okay. Apa lagi?
Okay. Assalamualaikum.
Assalamualaikum warahmatullahi wabarakatuh.
Terima kasih doktor.
Terima kasih.
Doktor.
Thank you, Doktor.
Terima kasih doktor.
Thank you, Doktor.
Thank you doktor.
Thank you, Doktor.
Thank you doktor.
Thank you, Doktor.
Thank you, Doktor.
Thank you, Doktor.